Hi, and welcome to my presentation on Chapter 4, Nucleic Acids, which are RNA and DNA. So nucleic acids are the next type of macromolecule that's commonly found in organisms that we'll be talking about. Nucleic acids are always used to work with hereditary information, and you have two basic types, DNA and RNA. DNA is used to store hereditary information, and RNA is used to work with it, to actually use it to build proteins or the other parts of the cell. Just like for proteins, the basic building block is the amino acid. For nucleic acids, the building block is the nucleotide. So we'll go over the structure of nucleotides and then get into the structure of the nucleic acids. So every nucleotide has three basic parts. It has a sugar in the middle, and then attached to that sugar it has a nitrogenous base and a phosphate group. What sugar it is depends on if it's RNA or DNA. RNA nucleotides have ribose as their sugar, and DNA nucleotides have deoxyribose as their sugar. And then there's 4 different nitrogenous bases that you could have. The carbons in the sugar are always numbered from 1' to 5'. They're numbered as like 1' to 5' instead of just 1 to 5 because the carbons in the nitrogenous base are also numbered, and so they get like 1 to 5. They get the 1 to 5 numbers, and so that you just add the prime sign to denote that that carbon is in the sugar, not in the nitrogenous base. So I have a couple diagrams of nucleotide structure. Well, on the top you have the actual structure of a nucleotide, and on the bottom you have the 2 different sugars that are possible, ribose for RNA and deoxyribose for DNA. So on the top here you can see your sugar is going to be in the middle, and then coming off of it on one side you have the nitrogenous base, and coming off of it on the other side you have the phosphate group. So the sugar has 5 carbons in it, and they start from kind of the right hand side, and it goes 1', 2', 3', 4', and then the 5' carbon is actually sticking up from the ring. The nitrogenous base is always attached at the 1' carbon. The phosphate group is always attached at the 5' carbon. And at the 2' carbon, you have basically the difference between ribose in RNA and deoxyribose in DNA, which you can see down here. So on this 2' carbon in ribose, you have a hydroxyl group, an OH, and on that same carbon in DNA, you have, or in deoxyribose, you have no hydroxyl. Instead, you just have a hydrogen. So it's called deoxyribose because it's like a ribose, but just with 1 oxygen removed from it. So you have 5 total nitrogenous bases that are possible to find in DNA or RNA nucleotides. They are adenine, thymine, uracil, cytosine, and guanine, or A, T, U, C, and G. In DNA, you can find adenine, thymine, cytosine, and guanine. In RNA, you can find adenine, uracil, cytosine, and guanine. So basically, they have 3 nitrogenous bases in common that you can find on their nucleotides, and then in DNA you can find thymine or T, and in RNA you find uracil or U instead. Every nitrogenous base is going to have at least 1 ring made out of carbon. It might have 2 rings also, so it's 1 to 2 rings made out of carbon, but some of those carbons are actually replaced by nitrogens in the ring, which is why we call it a nitrogenous base. And we can kind of categorize them into 2 different categories based on whether they have 1 ring or 2 rings. So the pyrimidines are the nitrogenous bases that have just 1 ring, and the purines are the nitrogenous bases with 2 rings. The pyrimidines are thymine, uracil, and cytosine, and the purines are adenine and guanine. And you can see them in this diagram here. On the top we have the pyrimidines, cytosine for DNA and RNA, uracil just for RNA, and thymine just for DNA. And then on the bottom you have the purines with their 2 rings, guanine and adenine, which would both be for DNA and RNA. So these rings are made out of carbon, but you can see at certain points there's nitrogen atoms inserted in there instead. So just like in proteins, when you have amino acids and you're trying to link them together to make a protein, you use a special bond called the peptide bond to link them together. With nucleic acids, you also use a special bond to link nucleotides together to form the actual nucleic acid. And that bond is called a phosphodiester bond or a phosphodiester linkage. The phosphodiester linkage is always between the 3' hydroxyl and the phosphate group of the next nucleotide. So you can kind of see in this diagram that phosphodiester linkage forming. You have two nucleotides here. You can tell that these are RNA nucleotides because they are ribose. They have the oxygen on the 2' carbon. On the 3' carbon of the first nucleotide, you have your hydroxyl, and that's going to have a reaction with the phosphate group on the next nucleotide, specifically with this hydroxyl that is part of that phosphate group. Once that reaction completes, you're basically going to have this oxygen that's on the first nucleotide attached to the phosphate group, and the atoms that are highlighted in blue are going to leave. And that would give you, of course, water, since it's an oxygen and 2 hydrogens. So once the reaction completes, now you have your oxygen that was attached to the 3' carbon on your first nucleotide, just also attached to the phosphorus atom of that phosphate group on the second nucleotide. And then you have your water that leaves. For DNA, in order to form a molecule of DNA, you have to link multiple DNA nucleotides together with those phosphodiester bonds or phosphodiester linkages. And if they're DNA nucleotides, that means that they have to have no oxygen on their 2' carbon in the sugar, so it's deoxyribose, not ribose. And then, of course, they would have thymine as a nitrogenous base instead of uracil. For RNA, in order to build a nucleic acid, in order to build a molecule of RNA, you have to get multiple ribose-containing nucleotides or multiple RNA nucleotides and link them together with phosphodiester linkages. And we always kind of say that the 5' phosphate group is kind of the front of a nucleic acid chain, and the 3' hydroxyl group is the end. So if you look at these kind of two linked nucleotides here, we would say that the top is the front and the bottom is the end. So on the top you have the 5' carbon and that phosphate group that's attached to it. So you're always going to have a phosphate group on the front end of its chain of DNA or RNA. On the back end, you're always going to have just this 3' hydroxyl group. So we say that the 5' end, where the phosphate group is attached, is the front end, and the 3' end, where the hydroxyl group is attached, is the back end. And that's because of the way that enzymes are able to link nucleotides together. You actually always have to start with a 3' hydroxyl, and then you can add the phosphate group of another nucleotide to that. You actually cannot start with a phosphate group and then add a 3' hydroxyl to it. There's no enzyme that can do that inside of the cell. So it always has to be added from 3' to 5', which means that on the front end, we're always going to end up with a phosphate group at the 5' carbon, and on the back end, we're always going to end up with a 3' hydroxyl group. That would be the place where you would be able to add a new nucleotide if you were going to keep extending the chain. So just like in proteins, you have the N-terminus and the C-terminus. In nucleic acids, you have the 5' end and the 3' end. So now we'll start talking about the structure of those nucleic acids, the DNA and the RNA, not just their nucleotides. So you start with the primary structure. For either DNA or for RNA, your first kind of level of structural organization is the primary structure. And it's basically very similar to the primary structure for a protein. The primary structure for a protein is the sequence of amino acids in the chain, and the primary structure for a nucleic acid is the sequence of nucleotides in the chain, or the sequence of bases in the strand. We can also refer to a nucleotide as a base. And that's done just because the most important part of the nucleotide is the nitrogenous base, or that's the part that can change from nucleotide to nucleotide. So all RNA nucleotides will have the same phosphate group and the same sugar, but they can have different nitrogenous bases, and the same thing for DNA nucleotides. We always write the strand as from the 5' end to the 3' end, so the 5' end is always the front, the 3' end is always the end. So on that 5' end, you're always going to have a phosphate group. On the 3' end, you always have a hydroxyl. If this is a primary structure for a DNA, then you're going to have A, T, C, and G nitrogenous bases. If it's for RNA, you're going to have A, U, C, and G nitrogenous bases. So the sequence of those bases, the A, T, C, and G, would be the primary structure for your DNA. The sequence of A's, U's, C's, and G's is the primary structure for RNA. So apart from those differences in the bases and the sugar that's found in the nucleotides for DNA and RNA, apart from that, the primary structure is actually going to be the same for DNA and for RNA. And so here you have an example of the primary structure where you have different different nucleotides, different bases linked together in this chain. So the sequence of the bases is what is determining the primary structure. And you can see you also have a backbone to this chain, which is just these phosphates and the sugars linked together. Always by the phosphodiester linkages between the 3' hydroxyl of one nucleotide and the 5' phosphate group of the next nucleotide. So the primary structure is basically the same for RNA and for DNA. But after that point, when you get into the secondary structure and the tertiary structure, that's going to be different for DNA and for RNA. So we'll talk about them separately. And we'll first go through the structure of DNA, which, as I may have forgotten to mention, is deoxyribonucleic acid. So the secondary structure for DNA is just the double helix. So you're going to have two strands of DNA nucleotides that are linked together with those phosphodiester bonds, and they're going to kind of come together so that their nitrogenous bases are linking to each other in the middle. So here you have an example of the double helix. In this case, it's unwound, so it's kind of straightened out for us. And we have one strand on the left, we have another strand on the right. The backbone, well, I guess you could say these are it's kind of forming like a ladder. The backbone is the phosphates and the sugars of each strand. And then the rungs of the ladder are the nitrogenous bases. And those nitrogenous bases will form base pairs with each other. Those base pairs are actually stabilized by hydrogen bonds. So all these dotted lines highlighted in yellow between the nitrogenous bases on each strand, those are hydrogen bonds between the bases. So that's kind of what's holding this secondary structure together, is those hydrogen bonds between the bases that form the base pairs. One thing to note here is that these two strands are not going in the same direction. So your first strand on the left is going from 5' to 3', so from front to back. But the strand on the right is going backwards. It's actually going from back, or the 3' end, to the front, or the 5' end. So this is an anti-parallel double helix, where these are anti-parallel strands because they're running in opposite directions. And they will always have to run in opposite directions. Then once you have those two strands coming together, linking together in the middle, using base pairs between their nitrogenous bases, then that whole structure is just going to kind of twist around itself, and that gives you the helix. Then the last thing I'll mention about this secondary structure is that, of course, those phosphate groups in the backbone are going to be charged. They have negative charges on them. So just like when you have a chain of amino acids, the backbone has charges on it, and those charges help keep the amino acid solubilized, or I should say help keep the protein solubilized. The charges on the phosphate groups in the backbone of DNA will help keep the double helix solubilized. So we'll just look in a little more detail at the base pairing. Again, it is just hydrogen bonds that form between two nitrogenous bases. Always you're going to have a purine paired with a pyrimidine. So it's always one nitrogenous base that has two rings paired with one that has just one ring. And it has to be that way, or else it's not going to fit within the space that's left, between the two backbones, or between the two the two strands that are forming the ladder. So there's just enough space between them for you to have those 3 rings and the hydrogen bonds in the middle. If you try to put two purines together, that would be 4 rings. It's going to be too big. That's going to force a bulge into the secondary structure. And if you try to pair 2 pyrimidines together, they just have 1 ring, and it's not even going to reach across. They won't be able to form hydrogen bonds with each other because they will be too far away. So the only way to create a stable secondary structure is if you have a purine paired with a pyrimidine, and then it will fit perfectly. And they're always going to pair in a particular way. So if you have DNA, you will always have thymine pairing with adenine and cytosine pairing with guanine. In RNA, it'll always be uracil that pairs with adenine, and then cytosine will still pair with guanine. And you can see those pairs here. On the top you have guanine and cytosine. On the bottom you have adenine and thymine. But if this was RNA, then we would have a uracil instead of this thymine, but the hydrogen bonds would be basically the same. So you notice that when guanine pairs with cytosine, it makes 3 hydrogen bonds, but when adenine pairs with thymine or with uracil, it only makes 2 hydrogen bonds. So because of that, the CG base pair is always going to be stronger than an AT base pair or an AU base pair. The other interaction that you have in the double helix that kind of helps hold it together is called base stacking. So when you have the nitrogenous bases in the double helix, they're actually going to be kind of rotated so that they're facing up and down. And you can see that in this diagram here. So your nitrogenous bases will always be oriented so that their flat sides are facing up and down, which means that they're facing towards the base pair below them and towards the base pair above them. And that is important to have so you can have the base stacking. When you have those nitrogenous bases stacked like that so that their flat sides are facing each other, they can actually interact with each other. We call that interaction base stacking. Actually, it's a type of interaction from chemistry called pi stacking. And we're not going to worry about pi stacking because understanding it just requires more complicated chemistry than what we are going to get into in here. So, so you don't have to worry about why this pi stacking occurs, but it is an interaction between 2 rings that are stacked on top of each other, but not just perfectly on top of each other. They actually need to be off kilter a little bit, like shown in this diagram. So you would have one and then on top of it, but off to the side a little bit, then you have the other. So in order for these bases to get into this arrangement so that they're not only just on top of each other, but kind of off to the side a little bit, they have to have the whole ladder twist around itself. So you would start with maybe a straight flattened out ladder, and then base stacking is going to cause that thing to twist around itself so that these bases can interact with each other and kind of stick to each other a little bit. So it's that base stacking that is actually causing the helix to form. So the base pairing between adjacent nitrogenous bases that are kind of across from each other, one on each strand, that base pairing is causing the strands to stick together and forming the ladder in the first place, and then that ladder twists to form a helix because of the base stacking. So that's the secondary structure for DNA. Then we have the tertiary structure, which is just how the DNA is packaged so it can fit inside the cell. So the DNA inside a cell is always going to be found in these large molecules called chromosomes that we had mentioned before. Every chromosome is really, really big. And you're going to have, some number of chromosomes in every cell. If it's a bacterial cell, it'll be just 1 chromosome, and it'll be, a bit smaller, but still really big compared to the cell itself. If we're talking about a eukaryotic cell, you're going to have a lot of chromosomes, and they're going to be very big. So in humans, you actually have 46 different chromosomes in every single cell. And they're all different sizes, but the biggest one is, if you stretch it out, is actually 8.5 centimeters long, and a cell is not centimeters long. And in fact, it's not just fitting in the cell, it's fitting in the nucleus of the cell. So a nucleus, how big that is also depends on the type of cell, but it's going to be usually between 6 and 10 micrometers across. And the biggest chromosome that has to fit inside it, if you convert it to micrometers, is going to be 85,000 micrometers long. So in order to make that fit, you're going to have to pack it really good. So the tertiary structure is just how you're packing that DNA up so it will actually be able to fit inside of a nucleus. You have kind of multiple layers of that packing, and you don't need to really worry about the details of it, but on the lowest level, you have your DNA double helix that is wrapped around proteins called histone proteins. So you're going to have like 8 of these histone proteins come together and stick to each other and make a little bead, and then you wrap the DNA strand around it. And that gives you what we call kind of beads on a string. After that, you're going to have those beads on a string kind of coiling around each other. Actually, let me just start talking about this with reference to my picture. So, you know, these little purple balls are the histone proteins. 8 of them will come and kind of stick together, and then you'll have the DNA wrap around that structure, and there you get your beads on a string. So these histone proteins stuck together is like a bead, and then once you wrap the DNA around it over and over again, that gives you beads on a string. Then if you coil that string up, you're going to get this coil here, which is actually, it has a name based on its thickness, but you know, we don't need to worry about that. Once you get these beads on a string coiled up to form this really tight coil, you're actually going to continue coiling it, and you coil it so much that you end up having supercoils. A supercoil is what happens when you coil something up as much as it can coil, and then you continue to coil it. This is probably not like, maybe not crazy relevant anymore, unfortunately, but it used to be that there was a very common example of a supercoil that everybody was familiar with in their day-to-day life, which was just the cord on a telephone. But of course, we use cell phones now. Some of you might not even have a landline, some of you might have one, but you know, if you've ever seen a corded phone, it looks like this. So you've got the cord of the phone coiled up. That's already as tight as it's going to coil. So when you continue coiling it, you get these supercoils, which are just coils of the coil, places where the coil is coiled around itself. So this type of structure is what's going to form in the DNA when you continue to coil it. So under normal circumstances, you kind of stop there once you get the supercoil formed. So most of the DNA that's inside of your nucleus is going to be at most packaged as a supercoil. But then if a cell is going to divide, then it's going to need to pack its DNA even more, and it's going to like really densely wind that supercoil up and kind of attach it to a protein framework, and that forms a chromosome that has the shape that we're used to seeing. So, you know, the traditional kind of squiggly X shape of a chromosome. That is the most packed form of the chromosome that you can have. That's only going to occur if the cell is about to divide. If the cell is not going to divide, then it'll just be in the supercoil form at the most. But actually, when you have DNA packaged into a supercoil, there's like no way to get up in there and use it for anything. So if you're going to actually use that DNA, that means you need to express some genes. That means that you're going to need to get in there so you can make an mRNA copy of the gene and then turn it into a protein. But you know, when you have it supercoiled, there's just no way for the enzymes that need to make the mRNA to actually access the gene. So when you have DNA packaged into a supercoil like this, it is basically inactive DNA. It is too tightly packed for anything to access it and work with it. So when you're going to actually express a gene, you have to unpack it. So you're going to have at any given time like part of the DNA in your nucleus supercoiled, and then part of it will be less tightly packed than that. Part of it will be more like just the coil or even just the beads on a string. To actually like get in here and read this DNA so you can build mRNA from it, you would need to even get rid of these beads on the string. You need to move the histones so that you can get to this part of the DNA that's wrapped around them. So that kind of, you know, that process goes beyond our purview in here, but Just know that, the more tightly packed DNA is, the more difficult it is to access, therefore it's going to be inactive. What your active DNA is going to be very loosely packed, so you can actually, work with it. So here you have a basic summary of the structural levels of DNA. First, you have the primary structure, which is just the sequence of nucleotides in the DNA strand. Then you're going to take two strands and attach them to each other to form a ladder to make the secondary structure, which, the secondary structure is just the double helix. So these strands are anti-parallel, so they're running in opposite directions from each other. They are held together in the middle by these bases, forming base pairs with each other, and those base pairs are just hydrogen bonds between the bases. You also have base stacking that then twists the whole ladder into a helix form. Then the tertiary structure is the packaging of the DNA. So first you're going to wrap it around histone proteins to get beads on a string. Then you're going to continue coiling that till you get a coil. Then you're going to continue coiling it until you get a supercoil. And then if the cell is going to divide, you're going to wind that supercoil up and attach it to a protein framework to form kind of the traditional shape of a chromosome that we're familiar with. And when it is loosely packed, then it is active. When it is tightly packed, then it is not active because there's no way to get in there and deal with it. So kind of when we're towards the end of the semester, we're going to come back to DNA and RNA, and we're going to talk about the process of gene expression. In gene expression, you are taking a gene and using it to build a protein, or using it to build, whatever that gene contains instructions for, but it's probably for a protein. So we're going to come back to this process of how you're actually working with DNA, and also the process of copying DNA in preparation for cell division. But we'll just go through kind of real briefly that process here, just a basic overview of it. So the information in the DNA is actually contained in the primary structure of the DNA. It is just the sequence of bases that has the information. So A, C, T, A, T, G, G, that's information in terms of DNA. That is how DNA is coding its information. So you don't have information stored in the secondary structure or in the tertiary structure, only in that primary structure, which means if you're going to copy DNA and you need to copy the information that's in the DNA, you need to copy the primary structure. Luckily, we have base pairing that allows you to copy the primary structure into a new strand. So whenever you're building new strands of DNA, you would start with your double helix, and the first thing you need to do is take that apart. So you're going to unwind the double helix and separate the two strands. Then you're going to get, on this left side, one parent strand, and on the right side, the other parent strand. So each of those two strands is going to become what they call a parent strand or a template strand. It's, the original strands, and each one of those is going to be used as a template to build a new strand. So you're going to have kind of free nucleotides that are floating around in the cell, in the nucleus, come in here and try to base pair with the bases that are sticking off of your parent strands. And they can only base pair one way. So if you have a C here, only this G can base pair with it. If you have another C, it doesn't work. If you have an A, it doesn't work. If you have a T, it also doesn't work. Only the G is going to fit in there. So if you have A, C, and T, and your primary structure for this parent strand, you're going to end up with T, G, and A in the new strand that you're building based on it. And if you look at your other parent strand, at that location you also have T, G, and A. So it's the same. The sequence of bases is going to be the same between the parent strands and the new strands that you're making. I guess I should say it won't be like literally exactly the same, but it's going to be what they call complementary. So it's complementary because A, C, and T can base pair with T, G, and A. There's a complementary sequence because they're able to, those two sequences are able to base pair together to form a double helix. So once you use each of these original strands to build a new strand, then you're actually going to end up with two new double helices, and each is going to be identical to the first one that you started with. So you have T, G, and A here, T, G, and A here. You're going to have your your A, C, and T here, and your A, C, and T here. I mean, I know the last base pair is hidden, but it would be in there. So the fact that these bases are only able to pair with each other one way is what lets you copy the DNA very easily to make new strands that have the same information as the old strands, and it also makes sure that you don't make any mistakes when you're doing that process, ideally at least. Okay, so that's the structure of DNA. Now we'll go over the secondary and tertiary structure for RNA, which I think I might have also forgotten to mention. The RNA stands for ribonucleic acid. So the primary structure for RNA, just to remind you, is the sequence of nucleotides in the strand or the sequence of bases in the strand. It's slightly different from DNA just because you have uracil instead of thymine, and because the sugar you have is ribose, not deoxyribose. But one thing that's really important there is actually the sugar. So ribose, when you make a nucleic acid using ribose, it's going to be a lot less stable than making a nucleic acid using deoxyribose. And it's because of this reaction that is kind of shown on the bottom here. So here you have a couple of RNA nucleotides. You can tell they're RNA because they have the ribose. They have the OH on the 2' carbon, so that makes this a ribose sugar. They're linked together using a phosphodiester linkage. So this would be like a little snippet of an RNA strand. What you can have, if you have a free hydroxyl group floating around in the cell, which you will do because you have water in the cell and it's going to kind of spontaneously react to form these free hydroxyl groups, you're going to have a reaction between this hydroxyl group and this oxygen. And you're going to have basically the hydrogen being stripped off of that oxygen to form water, and then you end up with a free oxygen that has a charge on it, and that oxygen is going to have a reaction with the phosphate group. That's a part of this phosphodiester linkage. At the end of that reaction, you end up with the phosphate group attached twice to the first nucleotide and attached 0 times to the second nucleotide. So basically you have broken the nucleotide chain. You've broken this strand of RNA. So anytime that you have RNA in water, basically that reaction is going to be occurring, and RNA will actually spontaneously degrade in water. If you have a strand of RNA and you leave it in water for enough time, it's going to chop itself up by reacting with that water. Until you get, tiny chunks, the strand will be left in tiny chunks and even individual nucleotides. So RNA is a lot less stable than DNA, and that's basically the reason why DNA is good to use for storing hereditary information, and RNA is not good to use for that purpose. It will not last. So the secondary structure of RNA is where it starts to really differ from the structure of DNA. The secondary structure in DNA is the double helix. The secondary structure in RNA is simple folding of the strand. So in DNA, for the secondary structure, you always have 2 strands. For RNA, you're just going to have 1 strand. Occasionally, you actually can have like double-stranded RNA, but that's really uncommon. Almost all RNA inside the cell is just 1 strand. So that strand is going to kind of fold back onto itself and base pair with itself to form the secondary structure of the RNA. And so it's a little bit similar to proteins. In proteins, the secondary structure is simple folding of the amino acid chain. Tertiary structure is complex folding of the amino acid chain. With RNA, the secondary structure is simple folding of the strand, and the tertiary structure actually is complex folding of the strand. It's still stabilized the same way as the DNA secondary structure, which is stabilized by hydrogen bonding between the bases. And by base stacking to make it twist into a helix. With the RNA secondary structure, you still have hydrogen bonds forming, or base pairs forming between adjacent bases, or bases that are kind of across from each other, that end up across from each other after the strand folds around on itself. Then once you have that base pairing, you're going to get some base stacking, and that causes little twists to form. So you're going to get little bits of helix, of what looks like double helix forming in the RNA secondary structure. So here's an example of a commonly found secondary structure in RNA. This is a single strand of RNA. Here you have the front end, the 5' end, and here's the back end, or the 3' end. And you can see the bases sticking off of it. So this is called a stem and loop structure. Here's the stem part, here's the loop part. Basically, you have two sections of this strand that are actually complementary to each other, which means that the bases would be able to base pair with each other in this part or these two parts of the strand. And they actually are doing that. They are hydrogen bonding together to form base pairs, and then you have the base stacking that forces this to twist and make the helix. So there's actually a number of different secondary structures that you can form in RNA. So for DNA, it only has one secondary structure. For RNA, there's actually several possible secondary structures. That ends up being important for the function of the RNA, which we'll talk about kind of later on. But yeah, the shapes that are formed in the secondary structure are important for the function of the RNA. These shapes will also kind of form spontaneously. So if you just have an RNA strand floating in the cell, it's going to kind of randomly like end up folding back on itself. And any place where you have complementary sections of that strand, once they get close enough to each other, they will, hydrogen bond with each other, base pair with each other, and then you're going to get the secondary structure forming. Since the secondary structure is going to be based on hydrogen bonding between, or base pairing between sections of the strand that are complementary to each other, that means that it's basically determined by the primary structure. So in order to have this stem structure forming in the RNA, you have to have these two sections of the RNA having a specific primary structure that's going to be complementary. So in this section of the strand, if you have a GCU, then and then in the other section of the strand you have to have a CGA. If you had a CCA, you're not going to form base pairs here, and this secondary structure isn't going to form. So it's basically entirely dependent on the primary structure being different parts of the primary structure being complementary to one another, so that they can base pair with each other to stabilize the secondary structure. Then, as I kind of mentioned, the tertiary structure for RNA is complex folding of the strand. It's still stabilized by base pairing between different parts of the strand, but here we're talking about parts of the strand that are kind of further away from each other than the parts that base pair with each other to form the secondary structure. And you're going to get kind of a lot of just bends that form in the strand as a result. So it won't just be like little sections of double helix, but it'll actually be like a larger 3D shape, which is going to be very variable. Kind of more similar to a protein. Every protein in its tertiary structure is going to form a unique 3D shape. With RNA, the shapes that you can make are not quite as diverse as the shapes that you can make with protein in the tertiary structure, but they're still very diverse. There's a lot of different shapes that you can make in the RNA tertiary structure. With DNA, you're only ever making a couple of shapes, right? You get your beads on a string, then you get your coil, then you get your supercoil, then if the cell is going to divide, you get the X-shaped chromosome, and that's about it. So every chromosome will always form those same shapes. For different molecules of RNA, they're all going to have a different secondary structure, and they're all going to have a different tertiary structure. So you can really form some more unique or unusual shapes. That ability to form a lot of different shapes is what lets RNA do its job. So DNA only has one function in the cell, just to store hereditary information. It's good at that job because it's very stable. RNA would be bad at that job because it's not stable, but it can form a lot of different shapes, which lets it have a lot of different functions, and a lot of those functions are going to be, I guess I should say, RNA has a lot of different roles to play in the process of working with DNA to actually build proteins. It's not just mRNA, but there's other types of RNA that are involved in that process as well, including, as we kind of mentioned, rRNA, that's part of the ribosome, that actually has the ability to perform catalysis. So in order to have all those different functions for RNA, you have to have all those different shapes that RNA can form. DNA would not be able to do all those different functions because it doesn't make a lot of different shapes. It only makes just those few stereotypical shapes. RNA has a lot of possibilities for the shape, which lets it function in a lot of different roles. So in the diagrams, you have just some views of the tertiary structure for RNA. I guess here you have an RNA molecule called tRNA, which is actually important for building proteins. So later on in the semester, right, as I had mentioned, when we get back into like how DNA is actually used to build a protein, we're going to get back into this tRNA, and we'll talk about what role it plays in that process. But here you have its basic 3D shape, which is formed by its tertiary structure. If you were to kind of flatten it out, it would actually look like this. So here you have kind of the secondary structure of that tRNA with, we have some stems here and some loops, but then in the tertiary structure, that whole thing is going to fold up to make this shape. And then we don't like to draw this all the time because it's complicated, so a lot of the time we just draw this cylinder with a bend in it instead. On the bottom, you have a couple of other RNA molecules that have their own complicated tertiary structures. So you can see that they have, quite different shapes from each other and from this tRNA on the top. So a lot of RNA molecules can make, I should say different RNA molecules can make a lot of different shapes in their tertiary structure, which is necessary for their function. So here you have a summary of the structure of RNA. So just like for DNA, the primary structure is a sequence of bases in the strand. Then the secondary structure will be different from the DNA secondary structure. The secondary structure for RNA is just the strand, the single strand kind of folding back on itself and base pairing with itself whenever you have regions of that strand that are complementary to each other in their primary structure. And that forms basic simple structures, simple shapes. Then in the tertiary structure, you have more complex shapes that form. It's still based on base pairing between different regions of the strand, but it's probably going to be parts of the strand that are further away from each other. And also you're going to have the whole thing kind of folding up or bending to make a more complicated 3D shape. So here you have examples of that. On the top, you have the primary structure for a strand of RNA, and it's just a sequence of bases. This would be the actual information in that RNA molecule. Then you have the secondary structure, so you have several little stems that are forming different regions of the strand that are complementary to each other and can base pair with each other. So you get some simple folds and simple shapes here. Then in the tertiary structure, you're going to have more base pairing between parts of the strand that are more distant from each other, and you're going to have the whole thing fold up to form a more complicated 3D shape. So again, we're going to go over RNA functions in more detail later in the semester when we come back to the process of gene expression, but RNA has a number of functions that are all involved with that process, or most of its functions are involved with that process. So you have mRNA, of course, that basically copies information in a gene from DNA and then takes it to the ribosome, and the ribosome will read that information in the mRNA and use it to make a protein. So that means that you're starting out with information contained in the primary sequence or the primary structure of the DNA. You're copying that information into the primary structure of the mRNA, and then the ribosome is kind of decoding that information and using it to build the primary structure of a protein. But you also have other forms of RNA that are involved in that process. So you have tRNA and rRNA. You also have some other forms like siRNA, which is small interfering RNA, or miRNA, that's microRNA, and also others. So there's actually a lot of different forms of RNA that are involved in the process of building proteins in one way or another. So some of them are involved in actually building the protein, like mRNA takes information to the ribosome. The ribosome also has to use tRNA to make sure it gets the right amino acid at each point. rRNA is actually responsible for performing catalysis and linking amino acids together using peptide bonds. But then siRNA and miRNA are involved with regulating the process of transforming mRNA into protein. So RNA can also have regulatory functions as well. So it has a lot of different functions. It can have all those different functions because it can have a lot of different shapes. So it's bad at storing information because it's not very stable, but it's good at doing a lot of different functions because it has a lot of shapes that are possible for it. DNA, on the other hand, only has one function because it only has one shape, where, it just has one secondary structure and then a characteristic way that it is packaged up in the tertiary structure. But it's very good at that job of storing information because it's very stable. It'll last for years and years and years. So in addition to the RNA that's used in our cells to work with DNA and transform its information into proteins, you also have other functions of RNA in other types of cells. So for instance, there's actually some viruses out there that don't have DNA storing their hereditary information. They would actually have RNA instead. And then you have a number of ribozymes inside of cells. And the most famous one is just rRNA, which is ribosomal RNA. So again, a ribozyme is any RNA molecule that functions as a catalyst, or that performs catalysis, which means that it's going to speed up a chemical reaction without actually participating in that reaction or being changed by that reaction. It just makes the reaction go faster. So if you have an enzyme that is a protein that's a catalyst, a ribozyme is an RNA molecule that's a catalyst. rRNA inside of the ribosome will actually catalyze the formation of peptide bonds between amino acids. So in a way, it's the most important part of the ribosome. And it's not even a protein. Ribozymes are kind of similar to enzymes in terms of how they work, and they're also similar to enzymes and other proteins in that their function is reliant on their shape. So if you have a ribozyme that loses its tertiary structure, it is no longer going to function. And the final idea that we will conclude with is the RNA world hypothesis. The RNA world hypothesis is just the idea that RNA came before DNA and before proteins in the evolution of life. So today we have, DNA storing our hereditary information, and then we have that being transformed into proteins, and proteins are doing work in the cell. But RNA actually can store information and can do work in the cell. It can store information because it has its primary structure, which is the same as the primary structure for DNA, and it can do work because it can form a lot of different shapes. So of course RNA is not as good at storing information as DNA is, but it does, it can store information, and there are viruses who use RNA to store information. So it stores information well enough to get by, especially for kind of a simple cell that's not going to live for a long time. So it wouldn't work for you because you're going to live for many, many years, but the average microbe does not live for nearly that long. And so some of them, like a virus, can get away with using RNA for their hereditary information instead of DNA. And of course, at the very beginning of life, we don't know what those early life forms would have looked like, but probably RNA could have actually evolved before cells properly evolved. And so we could have been looking at kind of proto-life forms that wouldn't look like anything that we see today, and that wouldn't necessarily last for a long time anyways. So these kind of proto-life forms we think might have been using RNA both to store their hereditary information and to do the different jobs in the cell. So both as DNA and as basically ribozymes that are doing work. So this is considered a hypothesis because, there's not enough evidence for it to be considered like a theory or anything like that. But it is a hypothesis that a lot of scientists believe is probably correct. And a lot of scientists kind of believe that RNA would be that missing link between chemicals and life forms. So in chemical evolution, you're starting with simple inorganic chemicals in the oceans of the early Earth, and they would have been reacting together to form more complicated molecules and ultimately to form complex organic molecules. And in order to get life, you need to have a molecule that can actually replicate itself. So RNA potentially would be able to do this. So if you had kind of chemicals reacting together to form nucleotides, and then those nucleotides reacting together to form rRNA, or not rRNA, but just RNA, you would have had information stored in that molecule, and if that molecule had the proper shape, it's possible it could even replicate itself, and would have been essentially a proto-life form in itself because it's able to replicate. So we'll kind of go through the process of copying RNA before we talk more about the evidence for the RNA world hypothesis. This process of copying RNA is really similar to the process of copying DNA. Very similar, in fact. But of course, in RNA, you have just one strand and not two strands. So in DNA, to copy it, you got to take those strands apart, and then each one of them serves as a template to build a new strand. When you're copying RNA, you only have one strand to start with. So that strand is going to be your template. And you would have that strand floating around. RNA nucleotides will come in and try to base pair with the bases on this strand. They can only base pair in certain ways. So if you have a C here, it can only base pair with a G. This A can only base pair with a U, not a T because this is RNA, so it has U instead of T. This G can only base pair with the C. It can't pair with the A or with the U or with another G, only with the C. So that means that if you have a particular template strand and you build another strand based on it, it will have to have a sequence that is complementary to that template strand. So that's kind of your first step. But notice that now we don't have another strand that is actually identical to our first strand. We have a strand that is complementary to that first strand. So now what we need to do is separate these two strands. So here they are separating, and then you can use this new strand that you just built as a template to make another new strand, which actually would have the same sequence as the template strand. So if you're using, as the original template strand, I should say. So if you're using this new strand to make a copy of it, or to make a new strand of RNA, you have the sequence CACUG. The only nucleotides that can base pair with those bases would be CUGAC. And if you look at the sequence of our original strand, I should say GUGAC. If you look at the sequence of our original strand, it is GUGAC. So now you've built an actual exact copy of that original strand. So that's the way that you would be able to copy RNA. When you're actually doing this process, you're going to have some enzymes involved, of course. So you're going to have these base pairs coming, these free nucleotides coming in and base pairing with your template strand. That just happens by itself. But then once they're base paired, you have to have an enzyme that's actually going to link them together to form a strand. So you need an enzyme that's going to actually form phosphodiester linkages between those new bases to make a new strand. Then you have to have some way of getting them across or apart from each other. Maybe you can have an enzyme that does that, or if you just have like kind of hot enough conditions, then it'll happen by itself. Then you have more free nucleotides coming in, and you need to have an enzyme that's going to create phosphodiester linkages between them. And it can be the same enzyme that created those linkages earlier in the process. But of course, if we're talking about an RNA world hypothesis, this wouldn't be an enzyme we're talking about, it would be a ribozyme. Today we use enzymes instead though, so we don't do this. Actually, our cells and eukaryotic cells do not copy RNA. They don't use RNA to build new RNA. If they're going to make RNA, they use DNA to make it instead. But viruses that use RNA for their genome, or to store their hereditary information, they actually may end up copying RNA from RNA. So they have special enzymes that allow them to do that process. But in chemical evolution, if we have a ribozyme arise that's going to copy itself, that's of course a ribozyme, not an enzyme, that would need to be able to catalyze these phosphodiester bonds between nucleotides. At the same time, that ribozyme would need to be serving as the template, so it would need to be the template strand, and it would also need to be catalyzing phosphodiester bonds between these new nucleotides to make a new strand. So we don't know of a ribozyme that can do that. Theoretically, maybe one could exist, but hasn't been discovered yet. So that's kind of probably the main piece of evidence for the RNA world hypothesis that is missing right now. We don't have a ribozyme identified that can serve as a template and as a ribozyme that copies itself at the same time. So here we have kind of a summary of our evidence for the RNA world. So first off, if you're going to have an RNA world, you need certain things to be true. First, you need to be able to make nucleotides spontaneously from chemicals that exist in the early oceans, or maybe in the early atmosphere. Then you need those nucleotides to be able to spontaneously arrange themselves into RNA. So you need phosphodiester bonds to form between them without an enzyme or a ribozyme to make that happen faster. You need it to happen, just spontaneously. And then after you've got that RNA built, now you need to make a ribozyme that can copy itself. So it has to act as a template, and it needs to catalyze phosphodiester bonds forming. So maybe potentially you could have kind of like two molecules of it, and one of them acts as a template and unfolds so it can serve as a template, and the other one might be doing the actual catalysis. But you would need some type of ribozyme that can be both a template and an active ribozyme. So those are the pieces of evidence that we need. We don't have them. We have done experiments where you kind of try to replicate the conditions in the early Earth's atmosphere or in its oceans. And in those experiments, we've actually shown that you can have nitrogenous bases spontaneously forming from inorganic chemicals. And we can also form ribose spontaneously from inorganic chemicals. But what we've not been able to do is link those nitrogenous bases with the ribose with the phosphate to make an actual nucleotide. So we can make the parts of the nucleotide, the nitrogenous base and the ribose. Actually, phosphate is already just an inorganic molecule by itself, so we have that as well. But we can't link those three things together to make a nucleotide spontaneously yet. Then if we turn our attention to kind of RNA that's already formed, we do have, RNA that catalyzes bond formation. So you have rRNA that catalyzes peptide bond formation. And we also have some ribozymes that can catalyze phosphodiester bond formation, which is the type of specific, catalysis that you need in order to have a ribozyme that builds itself. We've also been able to make ribozymes that are able to create nucleotides, but it's not, that doesn't super help us necessarily, because in order to get the ribozyme in the first place, you already have to have the nucleotides. What we've not been able to do is make a ribozyme that replicates itself. So we have a ribozyme that can catalyze phosphodiester bond formation, but not one that can also use itself as a template and make a new copy of itself. So the evidence that we have is just we can make nitrogenous bases spontaneously, we can make ribose spontaneously, and we can make a ribosome that catalyzes phosphodiester bond formation. Those are basically the strongest pieces of evidence that we have, which are not like super strong, and that's why this is a hypothesis and not a theory. But the idea is just, it's just too tempting to let go of, so we're still working on it. So if we kind of just assume that the RNA world hypothesis is correct, then we can kind of imagine how life would have began, starting with making that self-replicating ribozyme. So once you have the self-replicating ribozyme, it's going to start making more copies of itself, replicating itself. As it's making more copies of itself, it's going to make some mistakes. So, in general, you can only have like one base pairing with any given base. Like an A can only pair with a U if we're talking about RNA. A C can only pair with a G. So that means that whenever you're copying RNA into more RNA, it should always be kind of done perfectly. But the truth is that in real life, mistakes happen. And those mistakes become mutations when they're not corrected. So occasionally, this self-replicating ribozyme is going to mess up, and it's going to put maybe a C trying to pair with a C, and it'll just be able to kind of force that in there, and that'll get passed on. So now you'll have this new copy of itself that it made. Won't actually be a perfect copy. It'll be a little bit different. It'll have a mutation. Most of those mutations will not be positive, or else will just not affect it at all. But sometimes when you get a mutation like that, it's actually going to add new functionality to that ribozyme, so it'll be able to do more things. So eventually, if we get enough mutations and enough of the right mutations in this self-replicating ribozyme, we might end up with a new version of it that's able to not only replicate itself, but also can catalyze peptide bond formation. Or we could get this self-replicating ribozyme kind of making new copies of itself and including to continuing to replicate those mutated copies that it's making until eventually it makes a new ribozyme that can catalyze peptide bond formation, and it's just continuing to replicate that ribozyme as well as itself. So you might end up with two, one that just self-replicates, and another one that catalyzes peptide bond formation. So once you get a ribozyme that catalyzes peptide bond formation, which we do have examples of, well, of course, our RNA itself is an example of that. Once you get that, then you can build proteins. Once you've been able to build proteins, you're basically going to stop using RNA for catalysis, and you're going to start using proteins instead. That's because proteins are just better at the job than RNA is. So proteins can make a lot more different shapes than what RNA can make, and they also have different chemical properties. So every nucleotide has kind of the same set of chemical properties because they all just look really similar, but when you're talking about amino acids, they can have really different side chains from each other. So you have some that are nonpolar, you have some that have a negative charge or a positive charge. Lots of different properties are possible, and that means that you can make an active site that has different properties. It can be acidic or basic or hydrophobic or polar, anything that you need for a particular reaction. So that means that proteins are going to be much better at catalyzing a lot of different reactions than RNA can be. And at that point, you're going to have these proteins kind of just taking over and doing all of the reactions in the cell. Eventually, you're going to have proteins forming that can make carbohydrates and fats. Some of those fats can, well, it will not properly a fat, but some of those lipids would be able to form into a membrane, and then you could have a membrane forming around the ribozymes and the proteins, and at that point you actually have like an early cell, theoretically. I guess I should say potentially. The energy source for all of this would be probably deep sea vents. So this would probably be occurring deep in the ocean where you have these hydrothermal vents that are producing a lot of heat. And that could be your energy source for building this self-replicating ribozyme, and for the self-replicating ribozyme kind of replicating itself and making new ribozymes, and these new ribozymes making proteins and everything. When you're in the very, very earliest stages of chemical evolution, when you're just building amino acids and nucleotides out of inorganic chemicals, then it wouldn't have to be deep sea vents. It could be that the energy source is deep sea vents, but it could also be that these chemicals are kind of like towards the surface of the ocean and they're evaporating and going up into the sky, and you used to have a lot of lightning in the sky, like, you know, more than we have now. So they could have just gone up there high into the atmosphere and gotten like shocked by some lightning, and that's a giant source of energy. So that could have caused some of these inorganic chemicals to kind of fuse together to form nucleotides or amino acids, and then they fall back to the ocean in the rain, and would sink down to the bottom and, you know, continue to participate in, I guess, the chemical evolution of life. So none of these things that I just went over on this slide are like things that we know had happened. This is just a potential path that life might have taken.